Good morning, everyone. And I want to start out by thanking Drs. Becker and Dodd and Ebony for inviting me out. I'm really excited to be here for a variety of reasons, but especially because I think this meeting has the opportunity to advance the whole field of mitochondrial medicine, not just transfusion. I also want to applaud Dr. Becker for putting the patient first, and so I think that's important, and I'm glad to see the families here. So I'm going to give an overview, a broad overview of our work today with mitochondrial dysfunction, and as it relates to aging Alzheimer's disease, because that's really what we focus on in my laboratory, because I've been so dissatisfied with the Alzheimer's drugs over the years, and I've been getting into this field maybe about 10 years ago. So I'm hoping a broad overview offers an opportunity to meet new collaborators, and I hope it generates important information for the families today. These are my disclosures. So one question we can ask, and I'm sure some of you have seen this pinwheel before, is why do we age? And I'm not going to go through all the elements of it, but there's several different so-called hallmarks of aging. And the one that's important to me is this one here, is mitochondrial dysfunction. And as I said, this is really what my laboratory focuses on. So we're trying to understand mitochondrial dysfunction because we believe that mitochondrial dysfunction really is associated with next generation therapeutics. And understanding these targets is what's going to help us develop therapeutics in the future for a variety of age-related disorders, but also even maybe the primary mitochondrial disorders. So in my view, and what I'm going to explain to you for a few minutes, are some of the studies that we've done with repurposed drugs, computer modeling, sex hormones, some diets and dietary intervention, and then last but not least, the transfusion. And I want to be crystal clear, my lab is new at this, and so I'm hoping I can learn a lot more from everyone in this audience today. So there is a history for mitochondrial dysfunction in Alzheimer's, and it really started back in the 50s with Dr. Denham Harman, who proposed the free radical theory of aging, and he later modified this in the 70s to make it the so-called mitochondrial theory of aging because of the trying to understand really the importance of Ross in all of this. And then in 2004, Swerdlow and Kahn, Dr. Swerdlow is at, I think, the University of Kansas, really tried to combine mitochondrial dysfunction with Alzheimer's disease. And so this is the basis of my work that started a few years ago. Now here's another pinwheel. I got this from Mito-Canada. I was on the board of directors for 6 years at Mito-Canada, and I think it nicely shows how there's a lot of different types of mitochondrial dysfunction. There's also the so-called primary mitochondrial disorders, which I'm sure all the families know about here. So the primary mitochondrial disorders are genetically based, and they're depicted in this illustration by the dark green circles. However, the more common diseases that you're probably familiar with are the age-related conditions that you see in the light green circles. And so these are diabetes and cancer and even Alzheimer's disease. So mitochondria, of course, are the main driver of brain metabolism, especially most important for generating ATP, and we get a few ATP from the Krebs cycle and glycolysis, but we really get 36 to 38 ATPs from the mitochondria. And I'm sure most people in this room know that the main function of the electron transport chain in the mitochondria is for electron transfer and to create a proton gradient. So this is all coupled to the generation of ATP and complex 5. And I mentioned complex 5, and you can see the other complexes here, because in my lab we do spend a lot of time trying to understand the contribution of the protein subunits in each one of the complexes. Now this is a study that I did as a postdoctoral fellow in Dr. Mark Vanson's lab about 100 years ago. And this was done in collaboration with Dr. Sullivan over here, who is working with Steve Sheff, Dr. Steve Sheff at the time. So we used Pat's TBI model, and we found that TBI induced so-called deficits in synaptic plasticity as measured by long-term potentiation, an experimental paradigm to measure plasticity, or LTP responses, and you can see that in the middle part of the diagram here. And what we found is that that this application of this drug that many of you might know in the transplant field, cyclosporin A, actually helped to decrease the LTP deficits. And it's important because we also believe that this drug actually stabilizes the mitochondria because of its ability to modulate the mitochondrial permeability transfer pore. So in my lab, we've also attempted to do some computer modeling. This was started by one of my biomedical engineering students a few years ago, Chris Kodanik on the left, and he focused his master's thesis on modeling some aspects of the mitochondria. And then we were able to sophisticate our model with the more recent attempts of my postdoctoral fellow in Canada, Dr. Eni. And so she's a very bright person with a PhD in electrical engineering and a PhD in computer science, and she helped us really get this model to a very important level of sophistication. So one of the aspects of the model that we're using is a graphical user interface that you can see here. And the way the model is designed, it allows the user to ask important questions. So you can actually input your own data. And so one application is trying to predict OCR, or oxygen consumption rates. And I'll talk a bit more about that in just a few minutes and how we measure that in an instrument called the Seahorse. So we think that this model is a good way for scientists to ask scientific questions and try to input their own data. We're hoping to submit this paper next month in the Biomedical Engineering Journal. So the other thing I just want to bring up is that mitochondrial mobility is important, and we kind of alluded to this, but you know they have other functions besides ATP generation, and that of course involves motility. So my former research associate, Dr. Sabir, had studied motility, and this is just an example of one of the confocal videos he made to try to track what the mitochondria are doing. And of course this is an important aspect and mechanism not just for our studies in sensory neurons, but also trying to understand transfer and mobility in general. So synaptic plasticity, of course, and memory require energy. And what the studies have been showing so far is that mitochondria move to sites of high energy demand, the synapses. And there they can deliver not only ATP, but other molecules, small peptides, and other cargo, and like someone said earlier, information that's important for new memory formation. Other work in the lab that was led by another postdoctoral fellow of mine, Dr. Yelena Dvorževic, looked at sex differences. And you may or may not know, but about 2/3 of those with Alzheimer's disease are actually women. And we don't know why this is yet. And it can't just be due to longevity alone. So this is one of the areas that we work on in my lab. And in this study, she used 2 different transgenic mice, the 3xTG mouse and the TGCRD8 mouse, which are widely used transgenic, or I guess these days I have to say genetically engineered mice, right? I can't say transgenic anymore. Okay. So this study took a few years, and not only did we compare males to females, but we looked at a couple different regions, the hippocampus and the cortex, and we also looked at age. And we looked at 2-month-old, 6-month, and 14-month-old mice. And so each representation, each graph here is really a set of graphs. And the red block arrows show you those sets of figures that were really statistically significant. So overall, we found that females actually develop mitochondrial dysfunction early, much earlier than the males. And it seemed that the hippocampus was affected first, and then later the cortex. But overall, it seemed that the cortex is more susceptible to mitochondrial dysfunction. Well, why do we have sex differences? And of course, another possibility is mitochondrial DNA. And so most of you probably know that we inherit all of our mitochondrial DNA from our mothers, and so this certainly might have an influence, but another possibility are sex hormones. So in the lab, my PhD student, who's hoping to finish this summer, is studying sex hormones. And in particular, he's looking at estradiol, which is a form of estrogen. So in this study, he's been testing estradiol on different cell models, DRG neurons, primary neurons, and so on, to model the effects of amyloid beta deposition or application. And to make a long story short, we found so far that estradiol can improve OCR, it can improve some of the complex protein subunits in the electron transport chain, It can improve neurite outgrowth, and it also has influence on biochemical signaling involving important molecules like calcium calmodulin kinase 2, AMP kinase, which is the energy sensor, and PGC1 alpha signaling. And I mention all that because really this has an effect. The signaling pathway has an effect on mitochondrial biogenesis. So in another study we did in the lab, this involves a dietary supplement, creatine, that you probably heard about. And we gave these mice a diet of creatine for about 8 weeks. And we found a number of interesting things for learning and memory, but also mitochondrial function. Creatine, of course, is an over-the-counter product that bodybuilders use to rapidly build muscle mass. So we published a couple different papers associated with this work, and overall we found that it improved memory, learning, and OCR using a seahorse instrument, the XF24. And it also had a modulatory effect on this calcium calmodulin kinase to an NF-kappa B signaling axis. And I'll just remind everyone, the NF-kappa B pathway is an inflammatory pathway that we study, and we're interested in the relationship between mitochondrial dysfunction and inflammation. This is a study by another postdoctoral fellow, Dr. Ida Adlimigadam, who tested a repurposed cancer drug. This is a drug used for leukemia. And this was tested in astrocytes taken from young mice. So nilotinib is a tyrosine kinase inhibitor, but with my colleagues at Georgetown University who study dementia, there is benefit found in Lewy body dementia and Parkinson's disease dementia, and so we're trying to figure out how that works and why that works. So in this study, she used nilotinib and found in astrocytes, she found it also improved OCR, it improved the number of mitochondria as shown by the electron micrographs you can see in the bottom left, and it also improved the expression of some of the mitochondrial complex protein subunits as you can see in this Western blot. So overall this had an effect on one of the same pathways we've been talking about, the calcium calmodulin kinase 2, PGC1 alpha, and NRF2 signaling pathway. And this pathway is important also for improving mitochondrial function because it has an effect on mitochondrial dynamics, especially processes like fusion and fission, and also mitophagy. So there have been a lot of milestones over the years in transfer, transplantation, and transfusion that I think many people in this room know about. It really started way back in the 80s and has gotten accelerated by doctors like Dr. Walker and Dr. McCulley and Dr. Becker, of course. So it's very interesting to actually meet some of you people in person for a change. And so this is someone I just introduced you to a minute ago, Dr. Ida Adlimigadam. She was my postdoc. Now she has her own lab at Southern Illinois University in Springfield. And so in collaboration with Matrix Bio, and they have a representative over here, Kyle, we did our first study in mitochondrial transfusion. She couldn't be here today, so I'm presenting some of this on her behalf. So in the first step of this study that we published a few years ago, Western blot experiments were conducted in young mice and also old mice. And so the idea was to first look at the change over age in the expression of these mitochondrial subunits from complexes 1 to 5. And you can see that in every case, the Western blots are showing a decrease in the expression of these subunits as the mice got older. So we were interested in what might happen if we transfuse mitochondria, and that's what we did. And this figure illustrates a procedure that we used for mitochondrial transfusion. And basically, again, we had young mice and old mice, and we took the mitochondria from the liver of younger mice at 1 month of age and injected them into older mice using a catheter approach in the jugular vein. And you can see there were 3 groups of experimental mice where we attempted 2 doses, 10 milligrams per kilogram and 20 milligrams per kilogram, and also a control or vehicle dose in group 3. And so this was done once every 2 days for 2 weeks for a total of 7 injections per group. So what did we find? Well, this is one of the results is that we found an increase in the expression of subunit complex 2. And you can see that in the bar graph on the right. So if you compare the black bar, the control value, to compared to the 2 doses that we tried, we found an increase in the expression of this particular subunit, SH or SDHB. So complex 2 is interesting because it's really the only complex that is interacting with both the Krebs cycle and also the electron transport chain. So it forms a crucial link metabolically between these two pathways. So this is another study IDA conducted more recently at SIU, and this is also in collaboration with Matrix Bio. And IDA, in this case, used indirect telemetry to measure maximum oxygen consumption levels in these mice. And so mice were transfused 50 milligrams per kilogram with mitochondria. This was isolated for 2-month-old animals, and it was transfused into 2.5-year-old animals. And it was done for 6 weeks, twice weekly, for a total of 12 treatments. And this treatment resulted in a significant change in the oxygen consumption. So there was an improvement. Okay. So I'm going to go through this rather quickly. This is really the overall scheme for extracting mitochondria. In this case, we're calling them mitelets. This is a term that Tom Benson at Matrix, together with Eric Ballard at the University of Laval in Quebec, coined. And it's really from his work that he published in Blood back in 2014, and that's the fact that he discovered that the activated platelets actually release their mitochondria. So these are now referred to in our team as mitelets. So the idea in this study was to really extract these mitelets and transfuse them into the older mice to see if there's any benefit. So this is some work that Dr. Patel, I believe, is presenting in this poster today. You have in the back. And so just to give you a couple of snippets, there's an improvement in this transfusion with regards to improving the amount of bacterial load, and also the survival rate. So I'm sure Dr. Patel can fill you more in the details of his poster. And this is the overall scheme for Matrix for how this might translate to the clinic, and to extract mitelets, and then to actually grow in a bioreactor. And I know Doctors Becker and Tom Benson at Matrix are strategizing ways to utilize mitelets and mitochondria in bioreactors, because what really needs to be done is this needs to be done and tested in the clinic, and of course one would need larger volumes of mitochondria to be able to successfully do this in the clinic. So there's a couple of questions, a lot of questions to be answered in this field yet. I don't have time to go through all of these. What are the best sources of mitochondria? We've been using liver. I don't know if interesting to hear what Dr. McCulley had to say today about his work. Is how long are the effects? So far we've only been doing it short term. So I think I'll stop there. I want to thank Matrix for their collaboration and our funding from them, my other funding sources, more recent funding from the NIH, and past funding sources that showed you some of the data that I presented here today. So thank you for listening, and I'm happy to